Welcome back, ladies and gentlemen. Today we are going to look into the history and how the structure of DNA was determined. We're going to look at how DNA is replicated or copied, and we're going to look a little bit more at its function. Before we get started, please keep in mind that DNA stands for deoxyribonucleic acid. Nucleic acids are made of nucleotides, and those have three parts: a sugar, base, and phosphate. And we're going to look at how nucleotides are put together in long strings to make a DNA molecule and to code for proteins. In the early 1900s, scientists were aware that a molecule was being passed from parent to offspring, controlling how that offspring looked and functioned. However, they were not sure if it was protein or DNA that was involved in passing down this genetic information. Many scientists thought it had to be protein because cells are so full of protein and there's very little nucleic acid in a cell. However, in 1928, a man named Frederick Griffith was working with pneumonia strains in mice, and he determined that it's in fact DNA that is the genetic information that tells an organism how to function the way it does. So after 1928, the race was on worldwide for scientists to determine the structure of DNA. If we could determine the structure and we could manipulate this structure in different organisms, then we could control how those organisms function and make them work the way we want them to. And this is a big field in biology today. So the people that you see here on the screen were responsible for cracking the structure of DNA. We see James Watson and Francis Crick, and here you see Maurice Wilkins, and they were given the Nobel Prize in 1962 for determining the structure of DNA. Watson and Crick were model builders, and they built models of DNA based on other people's research, primarily Rosalind Franklin's research, and you see her here. Rosalind Franklin worked in Paris in the 1950s and perfected the art of X-ray crystallography, which is basically using X-rays to take pictures of molecules. Now, she was hired on in King's College in London in the 1950s, while this man here, Maurice Wilkins, was on vacation. And Wilkins was under the assumption that Rosalind Franklin was going to be his assistant, but when he returned from vacation, he found an updated lab and a person who was much better at his job than he was, and his PhD student was now working under her charge. So there was a lot of animosity between these two from the very beginning. So Rosalind Franklin, like I was saying, worked with X-ray crystallography, which means you shoot X-rays at a crystal, and those X-rays bounce off and hit a film, leaving dark spots. Now, these pictures could take months to develop and years to actually calculate where the atoms were in these molecules. You're not actually taking a picture of the molecule, but you're looking at how that molecule deflects these X-rays. So there was a lot of calculation involved, and there were no computers at the time to do this for people. So eventually, Rosalind Franklin took this photo, and this is photo 51. It's a very famous picture. Now, Watson and Crick had been building models for quite some time, and when they would build a model, they would call her in to look at it, and she could very quickly tell you if this model was right or not. So Watson and Crick actually got their models wrong several times, and the college that they worked for was embarrassed by this and forbid them to make any more models for a while. Eventually, however, they had come to King's College to get some advice on building a new model, and this is when Wilkins stole photo 51 from Rosalind Franklin and gave it to Watson and Crick. When Watson and Crick saw this picture, they had already done some calculations of their hypothesized molecules, and they knew what they were looking at when they saw this picture. The X here means that it is a twisted helix. We have some diamond-shaped patterns here that tell us that this is a continuation of the helix. It just keeps on going. And we have some missing pieces here, and here. The fourth band is missing, and this is where the helix actually crosses. So Watson and Crick, this meant a lot to them, and so they very quickly built the correct model of DNA and called Rosalind Franklin in to look at it. She's like, okay, yes, this is correct. This fits my research. But she was never actually given any credit for determining this structure, and not because she was a woman, but because she worked with X-rays so heavily and was very disappointed when they had to tell her, you have to leave the lab, you've been overexposed. But she was very passionate about this field, and it actually gave her cancer, and she passed away at 37. So she passed away before the Nobel Prize was awarded, and you cannot win a Nobel Prize after you die. So she was never given any credit, even though she was very deserving of credit for the Nobel Prize in her research. Since the late 1950s and early 1960s, DNA has been studied very heavily. Heredity is how traits are passed on from parent to offspring. Genetics is the branch of biology that determines how the DNA code actually codes for a trait. And a trait is simply a characteristic of an organism, be it a physical characteristic or how a protein functions. Now, these are some traits in humans that are controlled by one single spot on our DNA. Whether or not you have a widow's peak, and you can see here that I do have a small widow's peak. That is a dominant trait, and we'll talk more about those later. Whether or not you can roll your tongue, if you can do that, then you have the dominant trait. At least one parent gave you the code for rolling your tongue. The length of your second toe, if it's longer than your big toe, I believe that is a dominant trait. Whether or not you have free or attached earlobes, and you can tell that my earlobes are attached. They don't hang down in a lobe. All kinds of traits that are controlled by one single spot, but most traits are additive, and we'll get to genetics a little bit more later. So if we want to look at the structure of DNA, we have to remember that DNA is made of nucleotides, and nucleotide is a sugar, base, and phosphate put together. And if we put a lot of nucleotides together, then we can get a strand of DNA. Notice how we have the phosphates and sugars on the outside. That's going to make up the backbone like you see here. And our bases are turned in toward the middle. And you can see here that we always put blue to yellow, and we always put red to green. So A's always go with T's, and C's always go with G's. And we can make this a very, very long strand that has a lot of information in it. DNA is actually shaped like a double helix. You can see it's got two backbones here, and they're twisted around each other. And like I was telling you, the sugars and phosphates are on the outside, the bases are in the middle. So DNA has one function, and that is to carry genetic information, and that genetic information is in the sequence of these A's, T's, C's, and G's, and the order that they go together, very much like how we read a book, and the sequence of the letters determines what we see in our minds when we read. The cell sees DNA in a similar way. So the sequence of those A, T, C's, and G's is going to tell the cell how to make all the proteins that it needs to function. We'll look at that a little bit more later on in the semester. Keep in mind that proteins have different structures. The primary structure is just a chain of amino acids, and those amino acids interact in different ways. So sometimes they will form a helix, sometimes they will form a sheet, just based on how those amino acids get along with one another. That would be your secondary structures, your helices and your sheets. And if we put those primary structures with the secondary structures, then we can get tertiary structures. And those are kind of blobs. And we can put more than one of those tertiary structures together to get proteins like enzymes that can actually do some sort of function for the cell. So you can see here, the DNA is being read to make mRNA. That mRNA is going out to this ribosome to make a protein. DNA has different locations in a prokaryote. Pro means before, karyo means nucleus. So prokaryotes evolved before there was a nucleus. They do not have one. There is no bubble to contain this DNA within the cell. So it's just hanging out in the cytoplasm. It's also usually a ring structure. There's very few bacteria that have linear structures. However, Lyme's disease would be an example of a bacteria that does have a linear chromosome. Most bacteria have ring structures. And they also have these small rings called plasmids, and plasmids give scientists a lot of trouble because they often carry things like antibiotic resistance, which means that they can give the bacteria a trait so that it does not die in the presence of our antibiotics. So prokaryotes, again, do not have a nucleus. That DNA is arranged in a circle and hanging out in the cytoplasm of the cell. Eukaryotes, U means true, and karyo again means nucleus. So eukaryotes do have a nucleus to contain that DNA. So you can see here we have the cell, and we also have this inner bubble with the DNA inside, and that would be the nucleus where the DNA is held. And most eukaryotes are going to have linear chromosomes instead of circular ones. So nucleic acids are made of nucleotides. We can see a nucleotide here. We have a sugar, phosphate, and base. And we can string these nucleotides together by connecting a sugar from one nucleotide to the phosphate of the next. The bases are going to be in the middle, A and T are going to go together based on their shape. They have a spot for 2 hydrogen bonds. And C and G are always going to go together because of their shape. They have a place for 3 hydrogen bonds. Hydrogen bonds are very weak, and that's going to be important for replicating DNA. We have to be able to separate one side from the other fairly easily. So again, we have these sugar phosphate backbone. The bases turned in toward the middle to give us the double helix, and there's 10 bases per turn in a DNA molecule. There are 2 types of nucleic acids. We have DNA, which is deoxyribonucleic acid, and we have RNA, which is ribonucleic acid. And there are some differences between them. RNA does not use thymine. Instead, it's going to use uracil. And DNA is going to use a deoxyribose sugar, and RNA is going to use a ribose sugar here for that nucleotide. So here's your base pairing rules. If we have a strand of DNA, the A is always going to go with the T's, the G's are always going to go with the C's. However, if you're making an RNA molecule from DNA, the U will go with an A. The T is still going to go with A's, the G is still going to go with a C, and the C is still going to go with a G. We're going to focus on this part for now. We will talk about how RNA is made a little bit later in the semester. Now, these pairings were determined by a man named Erwin Chargaff, and we'll look at him a little bit more here in just a second, and he called them complementary bases. Watson and Crick used some of this information in building their model when they realized that Erwin Chargaff had determined that A's and T's were always present in the same amount and C's and G's were always present in the same amount. They used that to determine that these base pairing rules. You have this link in your PowerPoint if you want to try to build this DNA molecule later. But basically, I want you to kind of think through, once you see this nucleotide here, what's going to go with it? What's a T? So we should expect an A. It's an A, so we should put a T. A is going to go with T. It's always the same way. A with T. And this is a G, so we're going to put a C with it. C is going to go with G. And again, you can kind of see how these shapes line up together. You've got your 3 dots for 3 hydrogen bonds here. And we have our 2 dots for the 2 hydrogen bonds between A and T. So it says at this rate, you will need to work day and night for 50 years to transcribe the DNA in one human cell. We don't have 50 years to wait around. And in fact, your DNA can be made much quicker than this, about 1,000 bases per second. So when Erwin Chargaff determined that A and T were always present in the same amount, he was able to determine how much of one nucleotide you would find if you had a certain amount of another. So if we had 20% adenine, then how much thymine would you expect to see? Well, if A and T are always present in the same amount, if you have 20% A, then you would expect to have about 20% T. And this is not always perfectly on the dot exactly the same. There's always some free nucleotides floating around in the cell, so it might be a little bit off, but it's really, really close to the same. Now, if we have 10% adenine in the cell, then we can determine how much C or G that we would have as well. So if we have 10% A, then that means we would also have about 10% T. And that's going to add up to give us 20% between these two. And if we have 20% between A and T, then that means that we have 100% max. So we have 80% of the DNA left between C and G if we subtract that. Now keep in mind that we also, not only do we have G, but we have C. So we have to be able to split this 80% in half between these two. And so if we do that, then that means that we have 40% G and 40% C, based on Chargaff's rules. And again, it's not always exact, but it is really, really close. Here's a little chart to kind of help you keep in mind the differences between DNA. DNA is double-stranded, whereas RNA is single-stranded. DNA uses deoxyribose sugar as the sugar in the nucleotide. So this would be the deoxyribose. If this were RNA, then this little pentose sugar would be ribose sugar. And your bases in DNA are A, T, G, and C. In RNA, we have A, U, G, and C. So difference here in which base is going to be used. So like I was telling you about how DNA determines traits, it's the sequence of those A, T, C's, and G's and what order they're in that's going to determine which amino acid is going to be used in the growing protein chain. Proteins are not directly made by DNA. That means that this DNA doesn't change its shape. It doesn't become a protein, but it's going to hold the code for how that protein is going to be built. It's basically like a blueprint. And you wouldn't use a blueprint to build a house. You wouldn't actually construct the walls out of blueprints, but you would look at a blueprint to determine how to use materials in order to build that house. So it is a two-step process when it comes to building a protein. We have DNA that's going to be transcribed into RNA, and then that RNA is going to be translated into a protein. And you're going to see this a lot later on in the semester, so we're not going to spend a whole lot of time with it now. So just keep in mind that this is the central dogma of biology. We use DNA to copy and make another identical strand of DNA in the process of replication. When we're using DNA to make a protein, we first have to transcribe it into RNA and then translate it into a protein sequence. And we will get to that a little bit more later. Protein regulation for a cell is very important. A cell's life is a set of chemical reactions, and those proteins are involved in making those chemical reactions go smoothly. And if these cells don't have the right protein when they need it, then it can greatly influence that cell's life cycle, and we'll get to that a little bit more in a minute. So when we think about gene regulation, think about the word regulation as involved in hunting or fishing. If you buy a hunting or fishing license, it tells you what you can hunt, when you can hunt it, and how much you can possibly take. So gene regulation in a cell is very similar. Regulations are the rules on when to make a protein, the type of protein that's needed to be made, and the amount that is needed in that cell at the time. And this is important, and I'm going to use fish as an example of why gene regulation is important. So when you buy a fish and you bring it home, you don't just throw it into the tank. If you do, the fish can die. The reason for this is that fish have certain sets of proteins that work for certain ranges of temperatures. And if you just throw the fish in the wrong temperature, it will not have the time it takes to build those necessary proteins to function at that temperature, and it will not be able to survive. So when you bring a fish home, you take the bag, you sit it in the aquarium, and you let it gradually come to the temperature that's in the tank. This way, the fish is triggered to make the proteins that it needs at that temperature range, and it has the time to make those proteins. If you don't do this, then the fish, like I said, it can die because it doesn't have the time to make the right amount of protein. Gene regulation in a cell is very important. If a cell is making too much or too little protein or at the wrong time, then it can lose control of its cell cycle and it can start to reproduce continuously. Cells normally have mechanisms to tell where they are in relation to other cells within their tissue. A tissue is just a group of cells that's all trying to function to do the same thing. So if these cells can no longer tell where they are, or if they've just lost control of their life cycle due to mutation, then they're going to continue to reproduce. And these cells are going to mound up, and this is where you get a tumor. Now, not all tumors are deadly. Okay, some tumors are what we call benign. They can be taken out with something like surgery. But a tumor can become malignant if these mutations continue to accumulate. And this means that the cell can then break away from this tumor and go to somewhere else in the body and create a new tumor, kind of like a seed. So this is what we call cancer, ladies and gentlemen, and cancer is a very serious problem to Americans. It kills about 1 out of every 5 people in this country. It is the second leading killer after heart disease, and we've already talked about heart disease a little bit. So most of the time, people in America don't die from contagious diseases. I'm not saying that doesn't happen. Obviously it does, considering we have COVID going around right now. Obviously people die from contagious diseases, but most of the time, people die from genetic mutations that are already in their system. So there's many forms and many causes of cancer, but it all comes back to these genetic mutations that cause cells to grow uncontrollably. Within an organism, most cells have identical copies of DNA. And that's very important that every cell has the instruction manual it needs to make the proteins that it uses to function. So you might be wondering, well, why do we have different kinds of cells? Why do I have eye cells and heart cells and lung cells that are all responsible for doing different things and look entirely different? So cells are different based on the genes that they use, based on the proteins that they make. Some cells have small changes in DNA due to mutations, and accumulation of those mutations, again, can lead to things like cancer. Now, there are unique organisms out there in the world, like chimeras, for example. This is an example of a chimera here. This is an organism with two distinct sets of DNA. For example, if you were to have a twin and you reabsorbed that twin before you were born, then you could possibly have two sets of DNA in the same body. So this is an example of a bird that has done this. This is a cardinal that is half male and half female. And so half of this organism has a completely different set of DNA than the other half. And there are examples in the human world of chimeras as well, like I was saying, if you reabsorb a twin. So DNA between different species has variations to it. And we're going to look at all of these things a little bit more later on. Now let's get to DNA replication. So DNA has to be replicated, and you have to have the same copy of DNA from one cell to the other so that every daughter cell has an identical copy of DNA to the parent. Multicellular organisms like you and I and everything else that you can see with your naked eye are going to use DNA replication to grow and repair injury. If you were to cut yourself, you don't want that cut to last forever, so you need to grow new cells, and you want those new cells to be exactly like the old cells, so you have to have identical copies of DNA being made from the mother to the daughter cell. Now, unicellular organisms use DNA replication solely as a form of reproduction, and we will get to how cells reproduce a little bit later in this process called mitosis. Today we're going to just focus on how DNA is replicated in preparation for mitosis. DNA replication is a really complex process. We're going to break it down into two steps with two main enzymes, but there's really lots more enzymes that are involved than the ones that we're going to be looking at. So we're going to talk about helicase. Helicase is an enzyme. It's got ASE on it, tells us it's an enzyme, and it's going to run through the DNA strands and break these hydrogen bonds between these A, T, C's, and G's that are holding one strand to the other. So it's basically going to take that strand and pull it apart. Once this strand is separated from the other, then these nucleotides can serve as a template for the other strand. So we have this molecule, DNA polymerase. Again, we have ASE for enzyme. DNA polymerase is going to look at this template strand, and it's going to incorporate new nucleotides based on those base pairing rules. A is going to be paired with T, and C is going to be paired with G. So when we finish, the old strand is going to be identical to the new strand that we're making. Now, DNA is synthesized in a certain direction, but we're not going to worry about that for the purposes of this class. So DNA replication is going to start with one double helix, okay? Helicase is going to run through and rip these hydrogen bonds apart and separate these strands. And DNA polymerase is going to come in and put new nucleotides in based on our base pairing rules, A with T and C with G. So we can see that this old strand has been color-coded purple. And if we look, we have our two new strands that are identical to each other and our old strand. But we have one purple strand and one orange strand. This is to indicate that we have one old piece of DNA and one new piece of DNA. So we call this a semi-conservative process. Half is saved when we make a new strand of DNA. Old strand saved, and the new strand is added based on base pairing rules. Here I've given you some practice. If this is what your template strand reads, then what would the new strand look like? Okay? I want you to write down what the new strand would read using base pairing rules, A with T and C with G. If you need more time, please pause. hopefully you have written T, A, T, C, G, G, C, T, A, T, C, G, C. Okay? Now, this process is very accurate. DNA polymerase hardly ever makes a mistake. We actually have about 6 billion bases to our DNA, and only about 6 of those are miscopied when DNA is replicated. DNA is very long, and it needs to be coiled up in order to save space. And you'll notice this when you build your model, that as you twist it, it's going to shrink down, and that helps the DNA to fit inside of a cell. There's about 2 meters of DNA inside of a single human cell, and enough DNA in your whole body to span the solar system twice. Okay, it's a lot of DNA, and it needs to have somewhere to go. So number one, it's going to be coiled up in the double helix. It's also going to be wrapped around histone proteins, kind of like thread is wrapped around a spool. So the histone is like the spool and the DNA is like a thread. It's going to be all wound around those. It can be twisted up very tightly into structures called chromosomes. And we have 23 pairs of chromosomes in the nucleus of a single human cell. And yes, I realize that this is misspelled, but it was the best picture I could find. So chromosomes are going to be the structures that are passed down from parent to offspring during mitosis, and we'll get to that when we talk about the cell cycle. Chromosome number varies by species. So humans, for example, have 23 pairs of chromosomes in a cell. Chimps have 24 pairs of chromosomes in a cell, so that gives us a total of 46 chromosomes, and chimps a total of 48. This little plant that you see here is called an ophioglossoid fern, though it doesn't look like a traditional fern like you're used to seeing, but it is in fact a fern. And it has the most chromosomes of any other species on the planet, with a total of 1,260 chromosomes per cell. That is a lot. So a chromosome is a condensed packet of DNA. You see here a chromosome and its exact copy after replication. If we unwind this chromosome, it's made of a single thread of DNA that's been all coiled up. And DNA has these distinct regions called genes. And a gene is just a segment of DNA that codes for a protein. So if we think about this, then there's a lot of gene segments on a piece of DNA, and therefore organisms inherit a lot more genes than they do chromosomes. Chromosomes contain a lot of genes in them. I like to think of chromosomes like suitcases. The bigger the chromosome, the more genes it can fit. Let's see what I did there. No matter what species we're looking at, DNA structure is always the same. Nucleotides are used with sugars and phosphates linked to form the backbone of DNA, and bases are going to be turned into the middle of that double helix. A will always go with T and C will always go with G. That's the same no matter what species we deal with. DNA replication is always the same no matter what species we are dealing with. The same proteins are used and the same steps are used. The same code is going to be used for DNA nucleotides to code for a protein. We'll look at that a little bit more later. So these are some pieces of evidence that support the idea that all species are related. What is different between all species is the genetic information, the sequence of these A, T, C's, and G's. The order of these bases is going to determine how the amino acids are put together to make a protein in an organism. And that is going to be different from one species to another. Keep in mind that DNA polymerase is very accurate when it's putting nucleotides in during DNA replication. It hardly ever makes a mistake, but it does occasionally make mistakes, and we call them mutations. That DNA is going to be passed from parent to offspring. And the more closely related two organisms are, the more similarities we're going to see in their DNA sequence. The more mutations we see between two species or between two organisms, then the more distant their relationship is in time. So if we look at this chart, we're looking at the evolution of a protein called cytochrome C. Cytochrome C is used in aerobic cellular respiration by any organism that uses oxygen to break down glucose. All organisms that use aerobic respiration have the sequence for this protein. But that sequence is going to be different from one species to another. So if we look at the DNA sequence in horses and these organisms, the fewer mutations we see, the closer related those organisms are. So between the horse and the man, we have 12 differences. Between horses and pigs, we have 3. Between horses and chickens, we have 12 differences. So this tells us that the horse and the pig have less time between their evolution than the horse and the man and the horse and the chicken. If we look at the chicken's cytochrome C compared to the horse, pig, rabbit, and man, then we can see we have the fewest mutations between the chicken and the pig. This is not saying that pigs come from chickens or vice versa, but it is telling us something about their relationship in time. If we look down, then we can see between fish and these organisms, these differences are becoming greater and greater. And if we look at the yeast, we have a lot more differences than we do with the rest of these organisms. So this is telling us that the yeast probably evolved first because it has so many differences in its DNA. The tuna probably evolved next because there's so many differences between its DNA compared to these chickens and these mammals, but less so than there is in this yeast. So this is just telling us something about relationships between organisms. So similarity is determined by how many genes are shared and how alike those genes are. And this is just looking at one gene. We would have to look at a lot more genes in order to determine true relationships between these species. Keep in mind that there are variations between members of the same species. You are not exactly the same as your parents or your siblings. There are slight mutations from one generation to the next. So that's a good overview of DNA. Here are the resources that I used. Big things to keep in mind: DNA is replicated. Big things to keep in mind: helicase is going to be the enzyme that tears these hydrogen bonds apart, and DNA polymerase is the enzyme that's going to come in and link new nucleotides by putting a thymine with an adenine and a cytosine with a guanine. Okay, hope that was helpful in explaining how DNA is replicated.